Welcome to today's session on thyroid physiology. Today we'll be talking a little bit about the thyroid gland, what it does, how the thyroid hormones are produced and metabolized and used in the body. We'll even go into talking a little bit about thyroid function tests and correlating with clinical findings. So let's start with our first slide. So talk a bit about our thyroid gland. So it sits right here. Here we've got our trachea and our thyroid cartilage, and we've got our characteristic butterfly kind of looking thyroid gland. We've got our right lobe and our left lobe. In the middle, of course, this part's called the isthmus, and it has the largest store of hormones in the body. It produces thyroxine and triiodothyronine, so T4 and T3, and it's required for homeostasis. Its main role in the body is regulating metabolism, and it targets nearly every tissue in the body. So on the right-hand side, we zoom in into the thyroid gland, and we can really see the detailed histology of what makes up the thyroid gland. So the follicle is the basic unit of the thyroid gland, and it consists of follicular cells, the green color cells, and we've got our colloid. We've also got parafollicular cells that sits on the outsides of the units. Here we've got blood capillaries, and here we've got blood flow coming through them as well. So these follicular cells are very important that they trap iodine ions. So they trap iodine, which is a very important ion in terms of thyroid hormone synthesis, and they collect this from the blood and they trap this. These follicular cells are also very important for production of thyroglobulin, which is going to be a precursor for these thyroid hormones. Together, these two enter the colloid, where they're further developed and synthesized. And when they're ready, they come back into the follicular cells and get excreted into the blood cells, where they can go and be active in the body. So let's talk about the hypothalamic pituitary thyroid axis. So we've got our hypothalamus. This releases something called thyrotropin releasing hormone, which then acts on the pituitary gland, which then releases thyroid stimulating hormone. This then acts on the thyroid gland, and this releases our T4 and T3. So the ones we talked about, these are our thyroid hormones. These then, once it gets to a certain level, it starts to activate a negative feedback. So it tells the pituitary and the hypothalamus to kind of slow down on their release of TRH and TSH. And the cycle continues. When the levels get too low, the negative feedback diminishes, and the thyroid gland can continue to secrete these T3 and T4. And it's important that the thyroid gland is mainly regulated by its production through this hormone, TSH. It's a main governing factor. So we're going to talk about 4 steps in the thyroid hormone: how it's made, how it's secreted, how it's transported in our blood, and how it works. Let's start with how it's made. So we've got our follicular cells and we've got our blood vessels. So like we talked about before, the follicular cells trap these iodine from our diet that travels in the blood as iodine anion. So they take this up into the cell through this symporter, the sodium iodide symporter. This is an active transport, and it can be stimulated by TSH, which makes sense. When TSH is like, let's make some more thyroid hormones, they can encourage this iodine trapping. So here the iodine is trapping, and it enters the cell where it can be further used to develop thyroid hormone. This is an important rate-limiting step in the thyroid production process. So very important to note that. All right, let's continue on how it's made. So now that we've got the iodine, now these iodine needs to be incorporated into these thyroglobulins. So thyroglobulin is these tyrosine proteins that are going to be used. So when iodine and tyrosine is combined together with these thyroglobulins, they're going to be made as precursors for our thyroid hormones. So this is going to be all done through this enzyme called thyroid peroxidase, TPO. So we've got our thyroglobulin, and we're going to incorporate our iodine that we've just trapped. So now there's two things that we can do. There can be one attached to the thyroglobulin, making monoiodothyroxine, MIT, or there could be two, making diiodothyroxine, DIT. So MIT and DIT. There's one and there's two attached. So perfect. So next step is, so now that these thyroglobulins have been iodinated, they're now taken into the colloid. So they enter the colloid. So there's MIT and DIT. They've already got there. You can see it's attached, the iodine anions have been attached. Now then they can combine to create our precursors of T3 and T4. So can you see there's one iodine attached to there and there's two here? What would 1 and 2 make? It would make 3. This is where we get T3. So that 3 refers to that 3 iodine anions attached to that thyroglobulin. Next, we could have 2 and 2. So if DIT and DIT mix together, it can make T4, and that makes tetraiodothyroxine. So this is where T4 and T3 are kind of named after. These precursors are then stored in this colloid, so that inner circle of that histology slide that we looked before. So it's important to know that this is not its final form. These are precursors to that T4 and T3 that will be later used. All right, so now that we've made our precursors for T3 and T4, they're incorporated back into the follicular cell through pinocytosis. And then now they fuse with lysosomes, or here they call it proteases. When they fuse with lysosomes, it starts to release that T4 and T3 that now can be active in that body. Through this lysosome release, some of them do get converted back into that MIT and DIT, which can be deiodinated and then goes back into that cycle where it can be reused again. However, most of them, T3 and T4, will then be secreted as its final form. So you know how it had the precursors of T3 and T4 had that thyroglobulin attached to them, which was then released by these lysosomes? What happens to them? Well, some of them get released back into the blood as a byproduct, and we can detect these as a good and it can be a good indicator of the thyroid mass or TSH stimulation. So for example, if we had some sorts of thyroid cancers, we can use this thyroglobulin that's released into the blood as a good indicator of how the cancer is progressing, whether there's recurrence or remission. And we can use blood, track it in the blood to monitor its progress. So it's pretty interesting. So here, the detailed process again, the iodine's trapped into the follicular cell. Then the thyroglobulin is also produced in this follicular cells. They enter the colloid where they're mixed together to form DIT and MIT. DIT and MIT then combine together to form either T3 or T4, which goes back into the cell. And then so these are precursors. Then they fuse with lysosomes to become the active form and are secreted into the blood. So that's a little overview. So where does TSH play a role in all of this? So TSH can actually activate these receptors on these follicles, and they can stimulate this iodine trapping, so encouraging this process to speed up. They can also encourage thyroid hormone synthesis and release, and also affect the thyroid cells proliferation and differentiation as well. All right, so now that we've got our T3 and T4 in the blood, so how is it going to be transported? So T4 is the main secretory product. The thyroid hormone actually produces most of it as T4. Most of our T3 is actually derived from the deiodination of T4 in extrathyroid tissue. So T4 is actually going to turn into T3. So while the thyroid produces both and releases both into our blood, it's mainly going to produce T4 and release T4 into our blood. T4 has a longer half-life, 7 days as opposed to 1 day for T3. More than 99% of our thyroid hormones are going to be bound to a carrier protein. So it's mainly going to be thyroxine binding globulin. Another important one is albumin as well. And why are they bind to the carrier protein? The main role is to delay hormone metabolism. We want them to stay as much in the blood as possible so it can be used for a longer period of time. So these are the bound thyroid hormones. Now, the small percentage which are called free, these are the ones that are metabolically active and can actually do the functions that they want to do. So in terms of a normal body, our free hormones are going to be relatively constant. We don't want them increasing too much or decreasing too low because it can affect our body. Sometimes for some other causes, we can have changes in our carrier protein. So how do we keep free T4 and the same no matter how the carrier protein changes? Well, our body can compensate. So for example, we want our free T4 to remain the same. So if we have too many carrier proteins produced, for example, in pregnancy or in estrogen states, our carrier proteins can rise. And therefore, our body can compensate by increasing our thyroid hormone production so that more of it is bound and our free T4 remains constant. If, for example, a carrier protein becomes less, so it becomes reduced, and why might this be the case? For example, nephrotic syndrome, where you're losing proteins through your blood, through your urine, or malabsorption, or some malabsorption when you're losing protein through your urine, or systemic illnesses, it can drop your protein production. And therefore, our body might need to lower our production of thyroid hormone so that less of it is bound and our free T4 can still remain the same. So normally our free does not change, so our free T4, and it's going to be our bound that's going to be the one that's going to be changing. All right, so in terms of the mode of action, free T3 is going to be our biologically active one, as we said before. And most of this is going to be T4 that's going to be converted into T3. So as you can see, the thyroid gland releases T4 and T3, and this can be converted into T3 in peripheral tissues. Now, this process can be regulated by the tissue itself according to its needs. So if something is if it's more metabolically active and it requires more activity, it can encourage this process and have greater T4 converted to T3, and this can be used by the cells itself. Now, once this T3 kind of approaches a cell, how does it actually work? Well, first it binds to a receptor and enters the cell. And there's two kind of pathways it can go, the genomic pathway and the non-genomic pathway. So it can activate receptors in the mitochondria to encourage ADP and increase. Here it's increasing cyclic GMP and activating that pathway, encouraging higher metabolic rate. Another way is that it can activate nuclear receptors, and it can activate genes, which can then produce proteins, which can control energy utilization depending on the cell's needs. So now that we've talked about how it works, how do we get rid of these hormones? Well, T4 is more likely to be known as a prohormone. It's a longer half-life, and it's not as metabolically active as T3. Therefore, it has a few therapeutic advantages because we can give it as a dose and our body can convert it to T3 depending on its needs. So therefore, we can administer T4 thyroxine as a medication. So T4 can be converted to active T3, as we talked about before. So here it can be converted to T4s here. It's converted to active T3. Or it could actually be converted to inactive RT3. So it may be the body doesn't require as much T3. So our bodies then diverts the T4 into an inactive form. So it can be adjusted to the body's needs on which way it's going to go. These two are then further metabolized into inactive T2. During these deactivating processes, the iodine that's released, because they're removing the iodine during these processes, this can be further recycled to produce more thyroid hormones later. All right, a final slide that we want to talk about is the axis and how it can be correlated with our blood results. So whenever we do have changes in our thyroid hormone synthesis, our TSH and our T4. So T4 is much more prevalent in the blood, as we talked about before. T3 is converted from T4 in peripheral tissues according to local needs, and this is our biologically active hormone. Therefore, when we're checking it in the blood, it's more appropriate to rely on interpreting free T4, and that's why we look at free T4. TSH is also an important factor to know whether the problem is due to the thyroid gland or whether it's due to a higher up cause. TSH is the most, TSH and T4 have an inverse linear or logarithmic relationship. Therefore, when there is a small change in T4, there's going to be a very large change in TSH. And therefore, TSH is going to be more likely to change first, and we're going to detect these changes first before we start looking at T4. So therefore, whenever we order test, we look at TSH. If it's normal, we don't even bother looking at free T4 and free T3. So let's say we have a TSH and it turns out to be high. Okay, so a TSH is high. There might be a few different causes. So let's say a TSH is high, but our free T4 is low. So our TSH is actually trying to compensate and saying, hey, we need to ramp up our thyroid hormones. It's low in the blood. I can see it's low. We need to increase the production of thyroid hormones. But for some reason, the thyroid gland is not working as well as it should. Therefore, we call it a primary hypothyroid. If it's normal, this is called a subclinical hypothyroid, or it might be developing into a primary hypothyroid. So what are causes of primary hypothyroid syndromes? One is iodine deficiency, which is quite uncommon in developed countries. Another cause is Hashimoto's thyroiditis, which is an autoimmune condition, antibodies against our TPO. So remember TPO, thyroid peroxidase, is an enzyme during our thyroid hormone production. If there's antibodies against that, then it does make sense why there's going to be a hypothyroid syndrome. Previous radioactive iodine or neck surgery can also be a cause. Another one that's also not mentioned is euthyroid sick syndrome, where a systemic illness or major surgeries can impair functions. But for this one, it would more likely to be in a low TSH, which is what we've seen. So a low F3 T4. So whenever we have a high TSH and high free T4, it's saying that the thyroid hormone is producing a lot of free T4 because of the high TSH. So for some reason, the feedback loop is disrupted. So whenever we have high T4, free T4, we expect our TSH to be low. We expect our TSH to be, okay, that's too much, let's slow down. But for some reason, it's too high. So what are some causes? Maybe there's an interference with the blood result. So maybe something secreting TSH. Maybe there is a tumor in the pituitary gland. Maybe there's an ectopic tumor that's producing TSH. Or maybe we need to consider thyroid hormone resistance. So the body is not recognizing the thyroid hormone. There's resistance to the thyroid hormones. And therefore, TSH is trying to increase production to make up for this resistance. All right, on the other hand of the side, TSH is low. All right, there's still a number of causes, and we need to have a free T4 to further analyze what's happening. So let's say it's also low. If the free T4 is low, we expect our TSH to be increased, right? So if our thyroid hormone has a problem and it's not releasing enough hormones, we expect our TSH to be increased and give it that extra boost. But for some reason, our TSH is low as well. So that tells me that there's a higher up cause. So maybe there's a pituitary tumor, or maybe there was some pituitary surgery or radiotherapy that's impaired its function of the pituitary gland. All right. The other side is there is high free T4. So whenever there's high thyroid hormones, our TSH is going to be low. It's going to be saying, hey, let's slow down on this thyroid hormone that's being released. We're having too much. Let's slow down. So therefore, it's a problem with the thyroid gland. And for some reason, our thyroid gland is producing too much thyroid hormones. So what are the causes? It could be Graves' disease, which is an autoimmune condition where there's antibodies against the TSH receptors. So our thyroid hormones, our thyroid gland has TSH receptors present all over its cells. These autoimmune antibodies mimic TSH and activate these receptors to increase production and increase the thyroid function. Another cause could be there's a toxic nodule. So maybe there's a nodule on the thyroid gland that's producing hormones. Or thyroiditis, so inflammation of the thyroid gland can also release hormones. So that concludes the end of the thyroid physiology. We'll have additional sessions to go through on clinical pathologies of the thyroid gland and how we can diagnose, treat. And if you have any further topics or any further ideas, please comment down below. And don't forget to like and subscribe as well. All right, I'll see you guys next session. Bye.